We're just waiting for the laptop to wake up. So I am one of those genetic people, as it was described in that first session this morning. And the approach that my lab takes is really taking a genetic approach towards looking at ways that we try and engineer species for survival. While it's waiting to load up, I also want to acknowledge the Palawa people on the lands where the thylacine last lived in Tasmania, and the Bunurong people, which are the lands on which I work and live in Melbourne. And hopefully we'll also get some slides to talk about the science, which would be nice.
The laptop's just decided to take a nap, but I think we got it.
You get it?
I think so.
Yes. Oh, no, yes. All right. So we're all familiar with this statistic, I think, particularly in this audience, that Australia has the highest global rate of mammal extinctions of any country in the world. And with this level of animals threatened with extinctions in our country, we're predicted to have 1 to 2 extinctions per decade moving forward. Obviously, everybody in this room is working to make sure that that does not happen. And the other key thing here is this is drastically increased by adverse weather events, of which we are experiencing them more frequently, and they're also far more intense, the ones that we're having. And the horrendous black summer that we had a few years ago is a great example of this, as is your well-cued cyclone bomb, or whatever they're calling it, that you have happening here at the moment. And the thing that can happen in any single one of those events is obviously it can drive an entire species to extinction. Or there are cases where species don't go extinct from that event, but it reduces the population number of species so much that although we might be able to prevent that species from going extinct by managing very carefully the individuals in that population, they just simply don't have the genetic diversity to thrive once again and to reoccupy and you know, really take over and really recover from such a humongous event that has happened to them. And so that's what I think about all the time, is if a species goes extinct, does that have to be the end? If we've lost genetic biodiversity for a species, is that all that we can do? And the answer is no. We have genetic tools today that can prevent and reverse both of those things, and those are the things that my lab really focuses on. So when I got asked that question, what does it take to save a species, is we need more tools and more tools for more species. I want that toolbox in my garage would be fantastic, but for species survival would be incredible. So we need to get just a lot more tools, I think, under our belts. So the things that my lab works on is what we call the Next Generation Conservation Toolkit. So we're working on things like developing really good methods for gene editing some of our native animals, assisted reproductive technologies, things like IVF, induced ovulation, you know, the things that we have that are, you know, very well described for lots of livestock, humans, other species, are really sorely lacking in our native species. Also creating stem cells. This is that fundamental cell resource. It's a renewable resource that lives on forever. It can kind of immortalise species. It can immortalise species biodiversity. And through that, creating biobanks, which is something I'm going to talk about in another session this afternoon. But I'm a huge advocate for biobanking as well. And I think it's one of the single most critical things, if not the single most critical thing, that we should be doing in Australia. So all of these technologies have immediate conservation benefits. They're also essential for the bigger mission of my lab, which is working on the de-extinction of the Tasmanian tiger. From Vivek's talk before about having this really big vision that people can get behind has been an absolute game changer in the way that I've been able to attract funding to the lab to have all of the conservation outcomes that we're working on. And obviously bringing back a species that humans have driven to extinction is a conservation benefit in itself, but some of these species play absolutely critical roles in the ecosystem. Some of them are ecosystem engineers and are just absolutely critical for preserving species and the fauna and the flora of that particular area. And so, again, they can have major conservation benefits. So this has been the major focus of my group, is trying to bring the Tasmanian tiger back. But what I want to talk to you about today is this is really just the very tip of the giant iceberg of conservation tools that using this as a tool to drive the development of those technologies has provided. And through doing this, we've been able to attract millions of dollars of funding from biotech companies, really forward-thinking, visionary, very conservation-focused biotech that really want to drive some of these technologies and moving it forward. So I think it's opened up, certainly for us, a completely new area of funding that we can start to get some of this serious money and serious resources that we need to develop some of these things for our native animals. I'm so driven to try and bring the Tasmanian tiger back because of just how incredibly unique it was. It played such a critical role in that ecosystem in Tasmania, and we know that that ecosystem has been completely destabilised by its loss. And so for me, I'm really driven by trying to bring that species back and put that species back into that landscape. And there really is no other way for us to manage that landscape in its absence. It's just absolutely critical to put that species back there. And I could talk all day to you about why that is so critical. But I thought I'd give you a quick rundown on how the process works. This is how to de-extinct a thylacine in 30 seconds or less, because we have a very limited time frame here. But the first thing that we've been able to do and get really, really good at is sequencing DNA from museum specimens. We are exceptionally good at this now. We can do it for specimens that are hundreds of thousands of years old. With the thylacine, we've been able to sequence a telomere to telomere genome. This is sequencing it from one end to the other through all the hideous repeat regions and other awful stuff that we all have in our genomes. And we've been able to completely rebuild that blueprint for a thylacine. And once you've got that, you've got the instructions on how to build your thylacine. We use that then into extinction science to then look around in nature and identify the closest living relative to your animal that's gone extinct. We still can't create life where there is none. So where that animal's gone, we can't recreate cells from that animal. So we have to start with living cells from its closest living relative. In that process, we sequence the genomes of about 20 other dasyurid marsupials, telomere to telomere, which are now resources available to the whole community. So it has a massive benefit there. We identified the fat-tailed dunnart as the closest living relative to our species, and they're about 99.9% identical to a thylacine at the genome level. So then if you want to re-engineer your thylacine, you just change that 0.1% of the genome that's different, and then you've recreated your thylacine instructions, your thylacine genome, and then you can use standard assisted reproductive technologies, which are things we desperately need to develop for our marsupials, as well as a lot of our other native species, to turn that cell back into a whole living animal. So every single one of those has huge conservation benefits. And so that's what I'm really going to focus on for the rest of the talk today, is what we're actually doing on the conservation front with those technologies as they come to hand, and then how we're using that then to eventually get to this point of having a thylacine to put back into the landscape. So one of the first big advances that we've made is really perfecting the methods to make marsupial-induced pluripotent stem cells, or just stem cells for marsupials. This is really a critical resource that you need to have for any conservation program, I think, moving forward. These are cells that will live on forever, so they're immortalised cells. They're cells that can be used to undergo genetic engineering if you need to do something to change the genome of that species. Those cells can be used to give rise to whole other individuals if you want to clone. You can also turn those cells into gametes, so eggs and sperm, if you want to be able to generate other ways of reintroducing diversity back into species. So this has been a hugely important advance, was our ability to make these cells, and it works really well across a whole bunch of different marsupials that we've applied this technology to. And this was largely driven by Stephen Frankenberg, who's in the audience today, who's driven a lot of this work in my lab. So from having those stem cells, we were able to get an ARC linkage grant to set up Australia's first living cell biobank. So that's what I'll talk about this afternoon. As I said before, I'm a huge advocate for biobanking, but this provides a resource that we would never have to de-extinct anything again. You'd actually have living cells frozen down from species. We can freeze down today all the biodiversity that exists in Australia, and then we can make sure that we never lose any of that again. Any of those species that die, or a subpopulation, or some specific diversity in individuals, if you have living cells frozen from that individual, we'll eventually have the technology to turn that cell back into a whole animal that you could put back into the landscape to replace that lost genetic biodiversity. This is the only way that we can protect against further biodiversity losses in Australia, I think, is to set up this biobank, particularly from things like bushfire and flood. This is an absolutely essential resource. And I'm doing a lot of lobbying to try and get ongoing funding for this living cell biobank. It's based at Museums Victoria, and it's accessible to anybody that wants to get cells from particular species. Also, if you're going out and you're working on endangered species, we send you kits that you can just collect little bits of tissue, drop it into the tube. It goes into a post pack that we give to you as well. You just drop it in a letterbox. It goes to Museums Victoria. We will culture the cells and then freeze them down and make sure that that animal and that species is never lost. So if you can come along to the session this afternoon, I'd love to rant more about the importance of biobanks. So just really, really important, I think. The other thing that we've been working on, again, the brainchild of Stephen Frankenberg here today, is really working on novel methods to save our wildlife. This is just one example of the northern quoll. They're going extinct because they eat cane toads. It turns out that where mammals naturally coexist with cane toads, they mostly have cane toad toxin resistant. And it's one gene in the genome that can give you that cane toad toxin resistant. And even more impressive is it's just one single nucleotide, one single letter of code in their 3 billion base pair genome that makes you either susceptible to cane toads or resistant to cane toads. So we now have a big project to go in and we're going to edit that single allele. We've already done this in Dunnarts, our model marsupial species, and shown that it gives those cells, and even in the quoll cells, 6,000 times resistance to cane toad toxin with that single nucleotide change. So this is a way now that we can not only save the quoll from extinction, but then the quoll can actually eat cane toads because they're no longer toxic to it. So it can actually help control the spread of cane toads in Australia, which would be absolutely fantastic, I think. And this is not introducing anything that would not naturally occur. The quoll would eventually evolve that cane toad toxin resistance allele if it had another 50,000 years to live alongside cane toads, but we know that's absolutely not the case. Cane toads are such terrible force spreading across Australia. They'll be long extinct before they have the time to evolve this. So we can accelerate that process of evolution, save a species from extinction, and also hopefully help control cane toads. But these are the kind of genetic solutions that do exist that I really want people to deeply consider and think about how we incorporate these into our species survival programs. And lastly, when I go into museums and look at these drawers, and I've been doing this for years as we're sequencing thylacine genomes and things, instead of looking at this collection now as a representation of what we've lost in biodiversity that's gone, now we have the tools and the ability to get DNA from any one of those specimens, wherever it was collected from, whatever point in time, sequence that genome, understand what diversity or lost diversity looked like, and then re-engineer that diversity back into our living species today to make sure that we can reinvigorate them to really work on full species recovery for our planet. So hopefully I've convinced you that de-extinction science isn't just about trying to bring back mammoths, dodos, and the thylacine, and there is real importance in each one of those examples, but it's really creating a whole set of new tools that we need for conservation, preservation, but also species restoration that I think is really critical. And I also think that, with the world that we're facing today, with the biodiversity losses that we've already encountered, we absolutely have to embrace this technology moving forward. You have no choice. This is the only way we have to reverse that extinction vortex, to bring back lost diversity and to reinvigorate those species. So we were asked for key recommendations at the end of our talks, but I think I'm the only one who did it. So, but the biggest call out I want to have is really to not think of genetics as being this terrible thing or engineering wildlife as being something we should never do, but to start to really think about these new sets of tools, to embrace where you think that they really could have an important role in conservation, and to start to really talk about this technology and how we want to see it used in the conservation landscape. And obviously, huge advocate for more tools for more species. So that's the acknowledgements, and thank you very much.